Okay, in this video, we're going to go ahead and move on to topic 3 of the Unit 1.2 notes. In these notes, we're looking at all these different types of macromolecules found in living organisms. Previously, we talked about carbohydrates in topic 1, proteins in topic 2, and in this video, we're going to look at topic 3. So we're going to start learning about nucleic acids and what these guys look like and what they do inside of living organisms. So nucleic acids, these are also polymers, just like proteins, just like carbohydrates, which means that these molecules, these macromolecules, nucleic acids, these are gigantic molecules made up of millions, billions even actually, in the case of nucleic acids, billions of atoms all bonded together, forming these gigantic molecules. But really, when you look at these molecules, it's just a bunch of repeating subunits bonded to each other over and over and over again. Those repeating subunits we call monomers. Now, when we're looking at nucleic acid, the monomer is called a nucleotide. So the monomer, the repeating subunit found in nucleic acids, are called nucleotides, and you're just going to see, which is this piece right here, which we'll look at more closely in a second, but it's this guy bonded to itself over and over and over and over and over and over and over again. And then also in terms of the elements that we're going to see in nucleic acids, so just to continue reminding you guys, in carbohydrates, there's carbon, hydrogen, and oxygen, and that's it. In proteins, we looked at and saw how there's carbon, hydrogen, oxygen, but also nitrogen, and also even some sulfur in proteins. And now in nucleic acids, there's also carbon, hydrogen, and oxygen. I'll give you a hint if you haven't caught on that they all contain carbon, hydrogen, and oxygen. But in nucleic acid, it also contains phosphorus, which we haven't really seen yet, and also nitrogen. And so those are the elements, those 5 elements are what you're going to see in nucleic acids. There's 2 types of nucleic acids. There's DNA and there's RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. We'll talk about what these guys are and what's different between them in a moment. Obviously, I'm sure you guys have heard of both of these guys by now, especially DNA, I hope, but this is the genetic material inside of a living thing. This is what stores all the genetic information needed to program and build a living organism. And so the function of DNA is to be that genetic material that stores all that information. And it's actually has the information needed to replicate itself, which is cool. DNA molecules can actually, when cells are ready, they have the information needed to make copies of themselves. They're able to do that, and we'll talk about that much later this year. But really within this molecule is the information needed to build all of your proteins, the proteins that are found in your cells and inside your body. And your proteins are just a bunch of amino acids like we talked about in the last video. It's a bunch of amino acids bonded together in a specific sequence. And that information, to put those amino acids together to build these different proteins that do like a million different things inside of your body, that is all found in the DNA molecule. The DNA molecule has that information. And again, way later on this year, we'll talk about how that actually happens, how that information from the DNA is used to put those amino acids together. But that's where all that information comes from. And then all those proteins that are made, like I kind of emphasized in the last video, that proteins are what do everything. Like proteins are the things that are controlling everything being built and made and everything that's happening in your body right now, and a lot of other millions of important things. They're responsible for giving you all your traits and your characteristics. Like if you have green eyes, the reason you have green eyes is because there's a protein allowing that pigment to be made in your eyes. And that protein, the instructions to make that protein was in your DNA. Anyway, that's DNA, super important, right? And then RNA. RNA is actually a little bit different. It's sometimes, in some viruses, it's the genetic material for certain viruses, which we don't technically count as living things. We won't worry too much about that right now. But again, most of the time it's DNA that's the genetic material. But RNA does have a lot of other functions inside of all living things. They actually have a pretty large variety of functions compared to DNA. DNA is just sitting there storing all this information. But RNA actually has a lot of functions and is doing a lot inside of your guys' cells. Most of those things have to do with protein synthesis, which is the actual building of proteins, and we'll look at that later on this year. So they're involved in that, and also regulating gene expression, which means RNA has some RNA molecules, they actually control which genes in your DNA are being used or not used at any given moment. And we'll talk about that again second semester later on this year. But that's the two types of nucleic acids we're going to look at in these notes, and we're going to look at the structure of these molecules right now. So like I said, these are made up of nucleotides. RNA is made of nucleotides, DNA is made of nucleotides, and they're just going to repeat and bind to each other over and over and over again. So here's a nucleotide. You need to be very familiar with it. A nucleotide actually has 3 components you need to know. There is a 5-carbon sugar, which we call a pentose sugar. So this is actually a sugar molecule. And that sugar molecule is attached to a phosphate group and to what's called a nitrogenous base. Okay, those 3 things together makes up a nucleotide. So a sugar, a phosphate, nitrogenous base. That's what you see in this picture as well. A sugar, a phosphate, a nitrogenous base. That's a nucleotide. That's the monomer that builds DNA and RNA. Now let's start talking a little bit, and throughout these notes we'll kind of talk about the difference between RNA and DNA in terms of their structure. First of all, one difference I can point out right now is this sugar is actually different in RNA versus DNA. So the nucleotides that build DNA nucleotides, they do have a sugar, a phosphate, and a nitrogenous base, but this sugar is specifically a sugar called deoxyribose. So deoxyribose is the name of the sugar that's found in DNA nucleotides. In RNA, you're also going to find nucleotides with a sugar, a phosphate, nitrogenous base, but the sugar in RNA is going to be a slightly different sugar called ribose. So this is ribose sugar. They're very much the same, except for one small difference here in a functional group. But in RNA, it's going to have this sugar, and DNA, it's going to have this sugar. So that's one difference between them. The sugars, though, are both pentose sugars. They're 5-carbon sugars. They have 5 carbons. Those carbons, usually in organic molecules, scientists have a method of numbering those carbons, because sometimes we talk about specific carbon atoms in that molecule. So in these 5-carbon sugars, the carbons are numbered 1, 2, 3, 4, and 5. We call it has this comma, it means prime. So we call it 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Those are the 5 carbons that make up this pentose sugar. And the nitrogenous base you guys see is connected to the 1 prime carbon, the first carbon that's numbered here. And the phosphate group is connected to the 5 prime carbon on this sugar. It's not too important. You don't need to memorize to that specificity. But these numbers, you'll kind of see them come up in a moment in a few slides. But just to kind of introduce you to that, that's what those numbers are talking about. It's numbering those carbons. Anyway, another thing in these nucleotides, so there's the sugar, but then there's the nitrogenous base. And there's 5 different nitrogenous bases that exist. There is cytosine, thymine, uracil, adenine, and guanine. These are all nitrogenous bases that you might find in a nucleotide, like in this nucleotide right here. We usually refer to them by, letter. We cytosine is C, then T, U, A, and G. Now these 5 nitrogenous bases fall into 2 categories. There's nitrogenous bases that are called pyrimidines, and nitrogenous bases that are called purines. Pyrimidines, the difference between them is their ring structure. The nitrogenous bases that are called pyrimidines, they have a single ring structure. So cytosine, thymine, and uracil, those are pyrimidines or pyrimidines. They have 1 ring. Purines have 2 rings. They have a double ring structure, and that's adenine and guanine. And so like in this picture right here, this was a purine nitrogenous base because it has a double ring. But it's not always going to be a double ring. Like if I go back to this very first slide, here you can see that some of these nitrogens have a single ring, this one has a double ring, this one has a double ring. And so some of these are purines, some of these are pyrimidines. So pyrimidines are the ones with 1 ring, purines are the one with 2 rings. There is a mnemonic device to help you memorize this, because you do technically need to know which ones are which. The mnemonic device that I use, which if you want to come up with your own, feel free, but I remember it as cut pi and pure as gold. So like you would cut the pi, if you had a pi, you'd cut pi, and cut stands for cytosine, uracil, and thymine, and pi stands for pyrimidines. Those are the pyrimidines. And cut is 1 word, so that has 1 ring. That's how I remember it. And then pure as gold, the pure stands for purines. Purines are adenine and guanine, the A and the G. So pure as gold. As gold is 2 words. They have 2 rings. Hopefully that helps. But those are the nitrogenous bases. Now, what happens, which hopefully this isn't a surprise, is that in these molecules, the nucleotides are going to bind to each other over and over and over and over again to build these giant macromolecules, which are ultimately going to form DNA and RNA molecules inside of cells. And so these nucleotides, they are joined together by, you guessed it, dehydration synthesis reactions, which form new bonds between monomers. So we take this nucleotide, and then we're going to make a new bond between them. That's a dehydration reaction. Or on here they call it a condensation reaction, same thing. It results in the production of a water molecule. And so we call this linkage between them, between this nucleotide and this nucleotide, we kind of just broadly refer to this, between this phosphate that's in between this one and this one, and this connection between them, we call this a phosphodiester linkage. So a phosphodiester linkage, that's the name of the bond that's connecting nucleotides together. So you got to remember that. That's the name of the bond that's connecting nucleotide with the next nucleotide. It's called a phosphodiester linkage. But you can see specifically that new bond is forming between the 3' carbon. So this, the 1, 2, 3, the 3rd carbon in this pentose sugar. So the 3' carbon in the sugar. And the phosphate group of the next nucleotide. So in this nucleotide, the 3' carbon, and in this nucleotide, the phosphate group, they're forming a new bond with one another. Phosphodiester linkage. And that can repeatedly happen, and will repeatedly happen thousands, millions of times to build these giant polymers, which we call polynucleotides. So polynucleotide is a strand of nucleotides, which will ultimately be your DNA and your RNA. And so these strands of nucleotides, polynucleotides, these polymers, actually have 2 ends that are chemically different, which I talked about this. We saw this in proteins as well. When amino acids are linked together, the 2 ends are chemically different. And we say that the molecule then has directionality. Same thing with DNA and RNA, nucleic acids. These strands of nucleotides have directionality. On one end, there's a phosphate group that you're going to find. If you go to the very end of one of these strands, there's going to be a phosphate group, which is attached to the 5' carbon on the sugar. So we call this the 5' end, which has the phosphate group. And on the other end, if you go all the way to the other side of the strand, which could be hundreds, thousands, millions of nucleotides away, but on the other end of the strand, it's going to have a hydroxyl group attached to this 3' carbon on the sugar. And so we call this the 3' end. So this end is going to look different than this end. This end has a phosphate group, this one doesn't. This is called the 5' end, this is called the 3' end. So when we're looking at a strand of DNA or RNA, there's going to be a 5' end, and there's always going to be a 3' end. Now, if you're building these molecules, new nucleotides can only be added to the 3' end. So if we want to join more nucleotides inside your cell, if your cells want to join more nucleotides to this growing strand of nucleotides, it has to be added to the 3' end. You can't add it to the 5' end, which is also like proteins. They have amino acids, and if you want to add new amino acids, it has to be added to the carboxyl end, and it can't be added to the other end. Same situation here. So that's that. So now if we look at the actual 3-dimensional structure of DNA and RNA and kind of look at how they're different, and similar in a lot of ways. First of all, both DNA and RNA have what's called a sugar phosphate backbone, which is kind of like the sides of the molecule. A lot of times we say, specifically DNA, we say it's like a twisted ladder, and the sides of that molecule, even in RNA, but when you have a polynucleotide, the side of it is called a sugar phosphate backbone, which is just a bunch of sugars and phosphates alternating. So you have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That's the sugar phosphate backbone. And then you have these nitrogenous bases sticking out. That's the basic setup here. That's true for DNA, that's true for RNA. They both have a sugar phosphate backbone. Now, RNA is actually usually single-stranded, so it's usually just one long strand of nucleotides. It can be very long, some are shorter, some are longer. And so they only have one sugar phosphate backbone, though. It's one strand here with nitrogenous bases pointing out. Now, DNA, and this should sound super familiar, you should have learned this many times by now, DNA is actually double-stranded, which means it's actually 2 complete strands of nucleotides. So there's a strand of nucleotides and then a second strand of nucleotides, each with their own sugar phosphate backbone, and they're actually connected together via hydrogen bonding that occurs between the nitrogenous bases in the middle. So the nitrogenous bases like adenine and thymine or guanine and cytosine, they pair up in the middle of these two strands, the two strands that make up DNA. So we say that DNA is double-stranded, RNA is single-stranded. And then the double strand of DNA actually will form this very iconic helical structure, which is called a double helix. So these guys twist around each other. And so that's DNA, and that's what DNA is always going to look like three-dimensionally. Now RNA can take on a huge variety of different structures. So again, it's just one long strand of nucleotides, and sometimes that strand of nucleotides will hydrogen bond with itself and form some folding that occurs, kind of like a protein. It starts folding in on itself a little bit. Sometimes it's just very linear, and it kind of depends on the RNA molecule, but they can take on a variety of different shapes and structures, whereas DNA is always going to be this double helix. Now, when these nitrogenous bases are pairing with each other, whether it's in RNA, sometimes they find each other and hydrogen bond with each other, and in DNA, they're always bonding to one another. There's something called complementary base pairing that's going to occur, which means there's a certain way that they have to pair together, a certain rule. And so in DNA and in RNA, whenever you have a cytosine, it can only pair with a guanine. So cytosine and guanine are the only ones that can go together, because they can form 3 hydrogen bonds with each other. So they're going to form 3 hydrogen bonds, just like you see in this picture, cytosine and guanine. They're going to form 3 hydrogen bonds with each other, and they will always pair together. In DNA, you're always going to see them together. If you see a G, it's going to be hydrogen bond with a C, and vice versa. Now, in DNA, adenine is going to pair with thymine, and that's because they can form 2 hydrogen bonds with each other, but that's it. They only have 2 hydrogen bonds with each other, A and T. And so in DNA, you're always going to see adenine with thymine or thymine with adenine pairing together with 2 hydrogen bonds in the middle. But if this was RNA, and I forgot to mention this earlier, so that's my bad. In RNA, there is no thymine. So in DNA, you're going to see guanine, cytosine, adenine, and thymine, these 4 nitrogenous bases. But in RNA, you're going to see, you'll also see guanine, cytosine, adenine in RNA, but you won't see thymine. Thymine's only in DNA. RNA, instead of thymine, has a nitrogenous base called uracil, which, if I go back to this slide about the nitrogenous bases, I told you there's 5 of them. Well, 4 of these are found in DNA, which is cytosine, thymine, adenine, and guanine. And then there's 4 of these that are found in RNA, which is cytosine, adenine, guanine, and uracil. So in DNA, there's thymine with these 3, and in RNA, there's uracil with these 3. So only in RNA would you find uracil, and only in DNA would you find thymine. So going back to this complementary base pairing, if you have adenine, well, in DNA, adenine will pair with thymine, because those are the 2 that go together in DNA, and there is thymine in DNA. And there's no uracil. You're not going to see any U's here in DNA. Now, if this was RNA, RNA would be, you would see adenine, and you would see guanine and cytosine, but instead of thymine, it would be uracil, which is a different nitrogenous base. Adenine and uracil bond together, and they also form 2 hydrogen bonds, just like adenine and thymine. So those are the base pairing rules that occur when the nitrogenous bases are hydrogen bonding with each other, which you'll continue to see in a lot of these pictures. And then another thing to point out about DNA, since it is double-stranded, like I said, and it forms this twisted ladder-like structure, those 2 strands, each strand of nucleotides, if you remember, has a 5' end and a 3' end. Like I pointed out earlier, these 2 ends are different from one another. And that's true on this strand of the DNA molecule and on this strand of the DNA molecule. They have a 5' and a 3' end, but those ends actually run in opposite directions. So if you're looking at a molecule of DNA, which is going to be double-stranded, there's going to be 2 different strands. On each of those strands, there's going to be a 5' and a 3' end and a 5' and a 3' end. But those ends will always, those 2 strands will always be running in opposite directions. So one strand would have the 5' on one end going towards the 3' on the other end. Well, in the other strand, then, you're going to see the opposite. You're going to see the 3' end going to the 5' end. So we say that one strand is running in the 5' to 3' direction, and the other one's running in the 3' to 5' direction. Like they're going to be opposite. Okay, so I don't know if I made that overcomplicated, but the two strands are going to be running in opposite directions because the ends are going to be flipped. Okay, and because of that, we say that DNA is anti-parallel. So it is parallel. These two strands are completely parallel, but they're running in opposite directions, which if I had like a marker, right? Like a marker has like a cap on one end, no cap on the other. So let's say it has like a 5' and a 3' end, and so does this marker. It has a 5' and a 3' end. Well, DNA will never be like this, where those two ends are the same on the same side. It's always going to be like this. It's always opposite. It's going to be running in opposite directions. So it's parallel, but it's anti-parallel. So it's not like this where they're running in the same direction. It's anti-parallel. They're running in the opposite direction, those two ends. That's going to be very important next semester, but for now you need to know that. And this kind of summarizes the differences then between DNA and RNA. So there is a lot of similarities. They're both made of nucleotides, right? And they have this sugar phosphate backbone. RNA only has one because it's single-stranded. DNA has two because it's double-stranded. So that's one difference between them, single-stranded, double-stranded. Another difference was the sugar in the sugar phosphate backbone, and the sugar that's used to make the RNA nucleotides is ribose sugar, whereas in DNA it's deoxyribose sugar that's used to build the nucleotides, and so they're going to have two different types of sugar. And then a third difference is the bases, like I pointed out kind of late, but in DNA and RNA they both have cytosine, guanine, and adenine, but in RNA you're going to have uracil, which is not in DNA, and in DNA you're going to have thymine, which is not in RNA. So those are three big differences there. And then the very last thing in these notes is the fact that ATP, which is a super important molecule we're going to be talking about a lot. Hopefully it's a familiar molecule from freshman bio. ATP is this molecule that's used in cells a lot of the time to provide quick energy to all these different things that need to happen inside of your body and inside of cells on this planet. There's a lot of processes that require energy, and ATP is this really convenient molecule that can provide that energy quickly to all these things going on inside your body. And ATP, this molecule, which is called adenosine triphosphate, that's what ATP stands for. It actually is a nucleotide. It's a nucleotide. It has a ribose sugar. So it's like an RNA nucleotide. It has ribose as the sugar, and then adenine is the base. And then instead of having one phosphate, it has 3 phosphates. So that's the difference. So it's basically a nucleotide, but instead of 1 phosphate, there's 3 phosphates. But what those 3 phosphates is actually what creates this convenient molecule that provides really easy energy for the cell. This second and third phosphate, this bond between them is super unstable, meaning like this bond, it's a really high energy bond that's going to break really easily. And when it breaks, it releases energy that the cell can then use to do stuff with. And so that's why this molecule is so, why cells love this molecule, because it has this really unstable bond that's just waiting to break and release energy. And that's what happens. ATP, ATP is going to be hydrolyzed in your guys' cells, meaning like that bond, a hydrolysis reaction, there's a bond that's going to be broken. And that bond is going to be between the second and the third phosphate. And that ATP will then become ADP, which is adenosine diphosphate, where it only has 2 phosphates. And like I said, that third phosphate is going to be broken off. And when you break that bond, energy is released. And cells are doing this all the time. Basically, all cells on this planet are using ATP every second of every moment of every day. This is happening in all of your cells to provide energy for lots of different things that need to happen. But just worth pointing out that that's a nucleotide. Anyway, this was all about nucleic acids. Hopefully you know what these nucleic acids are, which are ultimately your DNA and RNA, and the structure of these nucleic acids, and how it's just a bunch of nucleotides bonded together to form these larger strands, which there are some differences between DNA and RNA that you need to know, but hopefully not too complicated. That's it for this video. I'll see you in the next video when we look at topic 4, the last topic of the Unit 1.2 notes.